Okay, great. Good morning, and welcome to this panel discussion, Adar RNA editing, unlocking new therapeutic opportunities. My name is Kane Akai. I'm one of the senior healthcare analysts here at Chardon, and joining me on the panel are four co-founders and or CEOs of companies that are pioneering the development of Adar-based therapeutics. With me is Daniel DeBoer, founder and CEO of ProQR, Dr. Paul Volno, president and CEO of Wave Life Sciences, Dr. Ram Ayar, president and CEO of Korro, and Dr. Jin-Billy Lee, associate professor at Stanford and co-founder of RNA. So I want to thank all of you for joining us today for this panel discussion. We began hosting this conference several years ago due to the increasing number of companies that were focused on the development of genetic medicines. And as advances in science and technology have increased our understanding of the epigenome, gene expression, transcription, translation, this has led to the emergence of new therapeutic modalities. I think most of our audience is aware now that Adar, adenosine deaminase, is acting on RNA, is an enzyme that naturally edits a specific adenosine on double-stranded RNA, converting it to inosine, which is then read as a guanosine base during translation. This results in amino acid alterations that it can also impact protein function, unlocking new therapeutic opportunities. I think the interest in Adar editing is certainly heating up, especially interest by big pharma. Within the past 2 years, we've seen new collaboration agreements between Roche and Shape Therapeutic, between Lilly and ProQR, between GSK and Wave Life Sciences. Korro's about to go public via a reverse merger, and RNA just came out of stealth mode. And the field is poised to enter the clinic with Waves 006, which they filed the CTA for, and we'll expect to see first patient enrollment very soon. So with all that, Billy, maybe let me start with you, and perhaps you can level set this discussion and help us understand why the development effort is now focused on designing a synthetic guide RNA to recruit endogenous Adar. And maybe we'll go back to the 2012 paper by Thornton Stafford entitled RNA deaminase conjugate selectively repairs point mutations. Now this came out at a time when I think it was largely overshadowed by all the buzz surrounding CRISPR-Cas9 gene editing. But perhaps you can highlight the key findings in this paper.
Yeah. Thanks, Kate. And thanks for having me on the panel. Great to be on this panel with you guys. Yeah, so I thought also it's very timely today to talk about this Adar. Adar is one of the key enzymes for RNA modifications. We call RNA editing because it's A2I or A2G change is read as something else. But most of the modifications, for example, adenosine methylation is still read as methylation. So really, it's a modification that we call editing. So today, this recognition of RNA modification is really very important. Just like many other modifications, the inosine is a really important cellular machinery for us to evade some of the immune activations. So there's really a lot of interesting biology going there as well. To address your question, so the field of Adar engineering has really evolved in very important ways. The paper you mentioned from my co-founder, Torsten Schaffost, really is considered to be the first paper to really show the engineering aspect of using Adar. Of course, in that particular paper in 2012, Torsten and colleagues were using the engineered Adar that can be conjugated with some antisense oligo to form the double-stranded RNA with a target mRNA. It really took the field quite a few years to get to a point that we together can show that endogenous Adar can be used. So, I just maybe briefly mention that, and I'm happy to.
Right. So let's fast forward to that 2019 paper you co-authored with Stafford entitled "Recruiting Endogenous Adars with Antisense Oligonucleotides." So help us understand how that further differentiated the idea that going after the endogenous was the way to go.
Yeah, so prior to this endogenous ADAR paper, the key paper by Torsten, by Josh Rosenthal, was a co-founder of Coral, really showed this engineered ADAR can work. But really, if you want to make this as a medicine, it's a two-component system, in a way sort of similar to the CRISPR system, right? Cas and CRISPR. So for the ADAR, once you have the kind of two-component system with the engineered ADAR and the antisense sequence, you typically would encounter a lot of potential off-targets because you sort of overexpress this enzyme ADAR. So that can create a lot of potential issues, although you can argue this is only editing RNA. So now with the endogenous ADAR, you don't have to use the second component. Then you become a one-component drug, and that is only the antisense sequence, and that can be in an oligo fashion. So that really makes this as a modality a lot more interesting, because now the specificity is really high. Our work and others showed the specificity is really close to 100%. Meaning if you want to edit a particular adenosine, that's the only adenosine you edit, pretty much, and in the RNA in a transient fashion. So that specificity has really become a key feature. But of course, the reason it took the whole field and it's 7 years to come from the engineered ADAR to endogenous ADAR is because it wasn't easy to recruit the endogenous ADAR and to do the work. So I think it's still kind of a work in progress as a field, and how we really increase the efficiency of editing and by recruiting the endogenous ADAR. Of course, the work from us and others, including work from people on the panel, really now show, and it's for certain indications, certain targets, we can really show very high efficiency as well. That really brings the field to a new level.
So Daniel, during the same period of time, ProQR, quietly in the back of the lab, was developing its Axiomer technology. Obviously, one of the first things is we've talked about before that I thought was interesting about ProQR. Certainly, gene editing has had some significant advances in humans, but talk about some of the advantages that RNA editing will always have.
Yeah. So first of all, thank you for organizing this panel. I think it's super exciting that the field today has progressed to the level that we can now have a panel with multiple companies working in the ADAR space. I think to follow up on Billy's introduction, I think the ADAR field came a long way in the last 10 years. Indeed, 10 years ago, the focus was mostly on engineered components that had exogenous ADAR combined to a guide strand that would be delivered into the cell as a whole package. We saw that development in the field, and in 2014, we came at this obviously from a different angle. We came at it from a drug development angle, and we thought, if we can develop an oligonucleotide that is the same as other oligonucleotides, but now can recruit the endogenous ADAR, that would be a real added tool that we can use in our toolbox to develop medicines for diseases that otherwise we cannot treat. So we started tinkering around with that in the lab, and back in 2014, we filed our first patent that with a chemically modified oligonucleotide, we could actually recruit endogenous ADAR. And I think today the field has really shown that across multiple different targets with high editing efficiency, both in vitro and in vivo, in mice, in non-human primates, we're able to reliably translate that science. And that really opens up the possibility to treat mutations that otherwise would be untreatable. Obviously before we could do knockdown with siRNA, we could do exon skipping to cut out specific parts of the RNA. But today we are able to really edit individual bases in the messenger RNA. And I think we as a field are just starting to understand what the possibility of that technology is for medicines that we otherwise couldn't reach. Now to answer your question on the difference with DNA editing, I think RNA editing has a lot of advantages. First of all, we're using an existing modality. So everything is known about oligonucleotides. We know how to make them. We know how they behave once you administer them. We know their safety profile. We know how they distribute, what the half-life is. So from a drug development perspective, these molecules are very much de-risked already. The advantage is that they do not need any external components. As Billy just elaborately explained, we don't need to bring in the machinery because the machinery is already in human cells. So in the analogy with CRISPR-Cas9, we don't have to bring in any foreign components like bacterial enzymes to actually make the edit. All we need to deliver is this single-stranded oligonucleotide. I think from a safety perspective, there are significant advantages because we know that these edits are transient, which means that if there is an undesired effect, you can take away the drug and the undesired effect would go away, which kind of lowers the bar for entering into the clinic. But also the transient nature of this editing allows you to go into diseases where the edit you're making is not intended to be there forever, like pain or infectious disease or other therapeutic areas where you only want a transient edit. I think amongst others, those are some of the advantages of RNA editing.
Okay, and we do have a toolkit from other oligo designs that have been built and are available for you, such as backbone modifications, base substitutions, GalNAc conjugates, or other conjugates. That's all great, but ADAR has its own peculiarities, such as a preference for 5' and 3' neighbors and off-target. So talk about how you're incorporating those learnings and those challenges in the designs of your guide constructs.
Yeah, so we've been at this now for 8 or 9 years, since we, back in 2014, first found that you could use an oligonucleotide to recruit endogenous ADAR. And most of this time we have used to optimize the technology, really understand the basic science behind ADAR editing, and understand the design rules. So what rules apply to the design of an editing oligonucleotide to recruit endogenous ADAR? There are quite a few challenges to overcome, indeed, to understand which chemical modifications were tolerated in each position, which would not inhibit ADAR editing, but would enhance the characteristics of these molecules in terms of cellular uptake and stability and so on. As part of that optimization process, we've collaborated closely with an academic group at UC Davis, the group of Peter Beal, who's a key opinion leader in the field of RNA editing, ADAR specifically. And for example, in a partnership with Peter Beal, we have found certain chemical modifications that enhance binding of ADAR affinity, of the ADAR complex, so enhance the ADAR affinity to that location. With, for example, the Banner space that we have in the EER, so in the central region of the molecule. But also we've been able to overcome some of the limitations of the neighboring 5' base right next to the target adenosine in the work that we've done with Peter Beal. So amongst others, we have filed patents on the protection of all of those optimizations, but also on the use of oligonucleotides to recruit ADAR. We have about 10 different patent families just on the platform of Axiomer, of recruitment of ADAR. And we think with all of these optimizations that we have now made, and that not only us, right, also others in the field have made, today I think the modality is really at a point that we can start exploring this in the clinic.
Well, great. So Ram, your lead is targeting alpha-1 antitrypsin deficiency. I think people appreciate this as a single point GDA mutation. It has both a toxic gain of function. The mutant protein is difficult to clear from the liver. Also has a loss of function. Its normal function to protect the lungs is lost with the reduction in the wild type. So why does this represent a good initial target for both the field and for your opera design platform?
Well, thanks, Kate, for having us here on the panel. Just as a brief introduction, because a lot of folks may not know about Koro, we are a privately held company at the current moment. It was started by folks that started DNA editing technology. So Ness Birmingham, who founded Intellia, Ali Bey Bahani, who is one of the board members at CRISPR. And so we have a deep knowledge, not just at the board, but also on the company in terms of how DNA editing has been utilized and all the challenges and all the nuances around developing a novel technology. I'll just touch back to a point that Billy said. This concept of A-to-I change has been around since 1995, right? Brenda Bass at UC Davis, who's now in Utah, identified this concept of unraveling of double-stranded RNA. And so it's taken the field quite some time to understand, and we've had the luxury of standing on shoulders of giants like Alnylam and Ionis to understand how you can convert this concept into a drug. The second thing I want to highlight before I jump into your question is that people approach this as a technology rather than what the applications are, and that's why I'll come back to alpha-1 in a second. When you think about human disease in general, and genetics being a big component of it, you have to think about it not just as single point mutations for rare indications, but also for large patient populations where the underlying thesis around genetics is very, very important. We've been developing drugs in the pharma industry for the last decade with the concept of genetic validation, and we've now, with the 4 people here at the table, added a toolkit that you can actually address some of the underlying disease manifestations. Long-winded way of saying you still need to prove out the technology, you still need to figure out where you have highest likelihood of clinical success, you still need to figure out, take that first shot on goal, and provide patient benefit is the most important piece. And that's where alpha-1 comes in, right? Alpha-1 provides a rare genetic indication, which is not so rare. It's about 100,000 patients here in the US, and a little over 100,000 patients in the EU and worldwide, where you actually can't knock down the protein and provide clinical benefit, and you have to return the patient back to the original, or at least the functional original state of what the normal protein does. And in this case, returning back and converting that adenosine, which is mutated into a guanosine, back to a guanosine. So in that context, alpha-1 is a fantastic indication to go after, because as I said, we stand on the shoulders of giants. Alnylam and Ionis have shown that you can deliver an oligonucleotide, among others, to the liver, and now that is expanding, where some folks on this panel have also showed recently. We can go into other tissue types. We know that we can edit that single transcript across all the cells, unlike DNA editing, where you can only do it in a subset, and provide a benefit both in the liver, where there's aggregation of this protein, but also the manifestation of this disease is in the lung. You provide enough of the protein to have an impact in the lung and prevent further clinical pathology. So alpha-1 in that context is fantastic, because within your first clinical study, you will know, 1, if this modality works, and 2, you can provide a fantastic option for these patients that doesn't exist anymore.
Well, great. So the crescendo for all of this, Paul, is the filing of the CTA for WAVE 006 to enter the clinic. WAVE has also pioneered a number of different chemistries. And really the toolkit, while it provides all of this options as far as the backbone modification, the base substitutions, and what have you, the real skill is how to utilize these. You know, you need to balance stability versus activity, deliverability, immunogenicity. So with all of that, Paul, talk to us about how you're utilizing the toolkit to, number 1, obviously advance quickly into the clinic and optimize the design of 006.
Well, 1, thank you for having us all. I mean, it's exciting to have a panel on ADAR RNA editing. And to your point, I think the time is right, the time's exciting, and it's exciting to be going into the clinic to do exactly, Ram, as you were saying, which is really providing some validation now for the translational potential of RNA medicines, not just as a technology, but as medicines. If we go back in history, and I appreciate you bringing it up, I mean, the field has really been pushing forward since the 1990s and really understanding how do you make oligonucleotides that could interact with proteins, that could do things beyond silencing, splicing, but obviously bring this field of RNA editing forward. I think the work we've been doing at WAVE now for over a decade has really been approaching it, to your point, Kay, on chemical optimization. And I think if we think about really the field translating, as we learned, and I think the reference to RNAi is a great one. I mean, we learned a lot about the early field of RNAi being an exciting modality, tractable to do something really important in medicines. But the key hurdle in turning it from a technology to a medicine was really understanding how do you utilize chemical optimization to create the efficiency where we can talk about it as a platform, meaning applying it from one target to the next. And I think the work that we've been doing at WAVE, principally in a ubiquitous way across RNA medicines, the advancement of our proprietary PN chemical modifications, and as we shared in R&D Day, the variants and how we continue to optimize that chemistry, as a number of my colleagues I know have noted in the past, it was a key stepping stone to being able to translate the potential of RNA editing now into a tractable, sustainable field. Our paper in Nature Biotech showed that now you could optimize chemistry to see editing efficiencies as we now get up to, not just 50%, but as we showed in R&D Day last week, up to 80% editing efficiencies on the transcript yielding protein. So I think if we think about this as a field where we can move it from a technology into a platform, and a platform that can unlock medicines, that's exciting. Where we're doing that today is in the field of starting with alpha-1 antitrypsin. So we filed the clinical trial application for alpha-1 antitrypsin. We'll be dosing the first patient in the fourth quarter, and we'll have data next year. And the key driver on this is to show that with chemical enhancements on the RNA guide strands now, the field of you doing GalNAc editing is great, so you can do subcutaneous delivery for alpha-1 antitrypsin. You can take GalNAc off and optimize beyond that to get to extrahepatic indications, as we shared with CNS editing. Again, editing efficiencies above 65% or higher. And so the ability to start thinking about the target landscape becomes highly attractive when we talk about specific editing, when we talk about moving beyond base editing. And I think that was an area, as a number of our panelists have talked about, where I think is a huge opportunity for the field of RNA editing, where we have reversibility as a feature, not a bug. You have the safety that you get with the highly specific edits without bystander edits, the ability to start thinking about prevalent diseases. And so one of the applications we shared last week was really about opening up this target universe beyond finding single point mutations, where, like in alpha-1, consequential disease, being able to demonstrate that you could fix that and see a healthy protein restore protection of the lung, see decrease aggregates in the liver and protect the liver. But the opportunity that we've shown that actually you can edit a transcript to stabilize it. So a lot of us think about this field also being mRNA editing, delivering mRNA into a cell to be able to generate a new protein. What we showed is the ability now to stabilize transcripts, so that you can actually build copy numbers of transcripts inside the cell. And therefore, just by using RNA editing, you can actually increase the expression of a protein. And we showed several examples last week where by upregulation, we could change phenotype. So we took a obese diabetic mouse and could show that by increasing that protein, so editing about 80% and seeing a twofold increase in protein, you could actually see corresponding weight loss. You could see insulin sensitivity, all by generating this protein secretion from the cell. So, stepping back and thinking about where the applications are and how we all should be thinking about RNA editing is not just thinking about rare monogenic diseases that we can correct, important applications, interesting applications, but the real opportunity we have sitting in front of us is how do we think about big prevalent diseases utilizing the benefits of RNA editing, again, with reversibility and safety, as we talked about, being key components of potential therapeutics. The future's bright. So our goal is, we've been demonstrating sustainability. We introduced, I think, 7 new programs last week in terms of applications of both correction and upregulation. We're going to push forward into the clinic to validate. So hopefully by next year, we're sitting here talking at this panel about the first clinical data in RNA editing and how do we translate that to open up the field. But I think what's great about having a number of companies working in this space means we get to talk about it. Fields talked about DNA editing for a very long time, and I think it's consumed a lot of the discussions in the narrative. And I think the opportunity we have as we think about new indications around RNA editing is pretty substantial.
And Daniel, let me circle back to you, because you've got some other targets that you're initially going after, and maybe describe why your initial targets are good for proving out the validity of your Axiomer platform.
Yeah, I think we have an embarrassment of riches in the target universe of that Adar can address. So there's indeed the bucket of the rare diseases with G2A mutations, like alpha-1 antitrypsin, which is a really attractive target that I think we'll learn a lot about from a few other companies in the field. Beyond that, there's other buckets. So we can introduce mutations, for example, that are known to have protective properties against certain disease, against cardiovascular disease, against Alzheimer's, against obesity, against whole range of different diseases. We can also use Axiomer or ADAR editing to introduce variants that, for example, break protein-protein interactions in a disease pathway. So pathways that cannot be targeted with small molecules, for example, we can treat with Axiomer. Now, with that, I think we're just starting to scratch the surface of what's possible. We have selected 2 targets for our initial translation to the clinic in-house, in addition to the 10 targets that we do in our partnership with Eli Lilly. For these 2 targets, we have picked targets that we can study in healthy volunteers. So these are variants that we introduce in a wild-type sequence, which means that we can run our first study in healthy volunteers, be able to measure target engagement in healthy volunteers. And we think there are significant advantages with that. First of all, recruitment is easy, cost is much more affordable, time to execution is much shorter. Plus, the data that we generate is in a very clean background. It's in healthy volunteers. There's no disease state that you have to take into account. So it means that from a translational perspective, going into wild-type background is very attractive for these first targets. Second, both of these targets are liver-originated disease, so it's diseases that are born in the liver, but can be expressed in other parts of the body, which means that from the delivery perspective, as Paul and Rem have just touched upon, it's relatively straightforward because we know how to get oligonucleotides to the liver, and therefore the delivery of these molecules is pretty much de-risked. For both of these indications, there's good biomarkers, so we can essentially, once we've treated these healthy volunteers, measure in blood samples, take biomarkers and measure if the desired pharmacological effect is taking place. And the advantage here is that these biomarkers are so well described that we can actually translate them back to the non-clinical data and test these exact same biomarkers along the way so we can connect it back and learn a lot from these translational steps. So our first program is called AX-810, which is going after cholestatic diseases. These are diseases where there's a high concentration of bile acid in liver, leading to liver fibrosis, and can only be treated with liver transplantation, which is a very high unmet medical need. Diseases like PSC and BA, we could go after with this. Our second program is called AX-1412, which is targeting the B4GALT1 gene. Here we are actually introducing a protective variant that has been that's rooted deeply in human genetics research. So this is a variant that's found in a population here in the US that is described to reduce your chance of developing cardiovascular disease by about 30%. We can essentially introduce that protective variant in people that don't carry it. And that's what we're testing in AX-1412. Both of them, I think, are really strong translational targets, but both of them are also real products when they succeed in the clinical translation.
Okay, well, let's shift gears here. Let me throw this question out to the panel. I know what we've talked about makes it sound like developing these ADAR therapeutics is going to be easy, but let me ask you this. What are the biggest challenges and unknowns that are really kind of hindering the development of optimized guide RNAs at this point? Billy, why don't I start with you?
I think it's a great point. Okay. In a way, I agree with the panel. This is kind of easier as a new modality because of all the other RNA therapy advancements in the past 20 plus years to get to us to this point. So that would enable many aspects of this ADAR modality. But of course, this is a new modality using ADAR as a machinery rather than RISC or RNSH. So for that reason, a deeper understanding of the ADAR biology, particularly for the engineering perspective, is going to be very, very important. ADAR has been a very small field. It's kind of interesting now. It actually attracts probably more industry attention than academic attention. Typically, it's the other way around. So I would say, there are dozens of labs only in the world that really study ADAR. My lab happens to be one of that. So I think there's just a lot to be learned, even about the ADAR biology. For example, we know that for naturally occurring editing events in a short hairpin fashion, the double-strand RNA being formed, the ADAR can somehow specifically and efficiently edit a single adenosine among maybe 10, 20 adenosines within that double-strand RNA. But why is that? What are the features that really drive that specificity and efficiency? My lab and others have made some efforts towards that. But I think there's still a long way to fully kind of understand what I call RNA editing code, if you will, in that regard. So far, of course, most of the prototypes, we pretty much design a perfect antisense, except that AC mismatch, which is kind of known to optimize editing. But in nature, you pretty much don't see things like that at all. There are many other irregularities in the double-strand RNA. How do we incorporate what nature has really optimized into this kind of engineered oligo or antisense sequence requires a lot of thoughts and effort. And obviously the chemistry modification will make some difference, but that really is unnatural in a sense. But from a sequence perspective, that's more of a natural sequence. There's a lot to be learned. I'm sure many people in the field, including people on the panel, the companies on the panel, are trying to optimize the sequence along with the chemistry so that we can really achieve high specificity and efficiency. I think there's just a lot to be learned.
Ram, how would you answer that?
So I approach it from a slightly different perspective than Billy. I've had the good fortune of touching 6 drugs that are currently on the market and 1 coming pretty soon that Novo just purchased my previous company. And for me, the most important thing is that we can play it on in mice, we can play it on in monkeys, but at the end of the day, we need to see what this drug does in man and have enough of an opportunity to learn from what actually happens in a human system. Because as folks know, from a cancer standpoint, we've cured many, many mice. But at the end of the day, some of those drugs, when they're eventually taken into humans, don't live up to the same expectations as they do in animals. And so we've been very thoughtful in doing a few things. The first one is all of our designs are based on both the human ADAR present, because there are differences between mice, monkeys, and humans from an ADAR enzyme standpoint. And then the second, in that same vein, is also looking at the human gene to make sure that that's where you're looking at from designing some of these compounds. The second component is we've spent a lot of time and money identifying the translational differences between the species. And I think that's, again, going to be a very critical aspect as to how you take some of these learnings from cells, test them out in rodents to understand the drug-like properties, and then eventually go into humans to see if this drug works or not at levels that you really need it to work for that patient population. And so we've shared data in monkeys where we've made specific edits into the SERPINA1 gene to show that we can achieve the levels of editing that we need in humans, which for us, we believe in alpha-1 is above 50% at trough levels, and we've demonstrated that we can actually do that. That stuff we've done, right? Some of the challenges now are going to be, unfortunately, we're looking through the window of this machinery that happens inside a cell, and we're making judgments as to what's actually happening within a cell. I'll give you an anecdotal example. Frank Rigo and folks at Ionis took nusinersen and changed 1 different chemical modification on nusinersen, which eventually is Spinraza. That changed it from a splicing agent to a skipping agent by just making that 1 specific change. Right? So I would say that there's still a lot we need to learn, and this is for a modality from an RNAi perspective that we know a lot about how it engages risk, how it loads into Argonaut, et cetera, et cetera. So I would say it's still an evolving field, but the only way we will learn is if we go to humans and understand what it actually does in patients.
Paul or Daniel, anything else to add?
I think it's a great transition because I couldn't agree more. We've focused from the very early days, I said now over a decade, on learnings about how do chemical modifications translate. And as you pointed, some may remember that our firm foundation at the beginning of Wave, kind of the footprint was understanding the chirality of oligonucleotides. We talk a lot now about medicines, that's really important, but the root is really understanding, to your point, each individual drug. So the foundational part of Wave, when we talk about an AIMer, which is our RNA editing construct, it is a single characterized oligonucleotide, not a mixture of multiple oligos because of the modifications that get used. So from the very beginning at Wave, we've seen that having 1 drug that you can characterize preclinically, safety, take into patients, teaches you things. It teaches you things about how do you make chemical changes as you go. And I agree with Ram. When we moved into ADAR and started with our first Serpent A1 model, we actually created the human ADAR mouse model to look at identifying, can we edit? Can we see substantial in vivo editing? Does that translate to substantial protein? So again, not just 11 micromolar, but well above, like 20 plus micromolar of protein. And could we see that translate? And then we got a lot of questions. Everybody goes, well, but that's the human ADAR mouse. Like, what about the other one that doesn't have it? And we showed we could do that too. And so I think to this point, translation understanding, because the next iteration is by being able to chemically optimize, we can use GalNAc. So when we think about being able to target hepatocytes subcutaneously, deliver, actually what we have learned as a field, not just the ADAR field, but as an RNA medicines field, is there's a high propensity of translation between mouse to humans in terms of GalNAc conjugation translating. It's not to say that we don't do the concentrations in non-human primates. We do that. But what we can do is really lean on that translation potential. So as we built the alpha-1 antitrypsin program at Wave, the key was very much to Ram's point is leverage uniqueness in chemistry. That, I can tell you, drives huge ability to your point. When one thinks not just about engineering the protein, the paper we published in Nature Biotech showed that if we took a stereorandom guide strand for ADAR, it had a very, very low affinity rate in terms of engaging the enzyme and actually doing productive endogenous editing. It wasn't until we made, one, we controlled the stereochemistry around it to get specific interactions with the enzyme, but critically important was our PN chemistry modification. And I know others have talked about now the application of that, but when we took our proprietary PN chemistry and the variants, that's where the translational potential of ADAR was fundamentally different. It gave us potency. We've seen this now not just in editing. We showed durability in CNS silencing where we had 6-month windows where we were looking at knockdown. We've seen it in splicing. We received more exon skipping with our exon 53 program than anybody else in the exon skipping space. And I think this chemistry, as we're applying it in cells, is really driving a major therapeutic transition. And so as we take that forward, the last piece is exactly what Ram brought up, which is it's about human clinical translation. So I think we're really poised now to see the first RNA editing medicine potentially translate, hopefully translate. But I think as a field, we're going to learn a lot about it. We're going to learn about characteristics on the other fields of translation, from what in vivo models translate well to human models. How do we think about editing efficiency in terms of protein production? And these data that we'll have next year will be about protein production. We'll be able to do mass spec analysis on the protein so we can see, can we recapitulate what we saw in the animal model of that transition from a Z protein to a wild type protein. And we're going to learn a lot about pharmacology. So I think it's going to be really exciting for the field of RNA editing.
All right, well, let me take advantage of the collective brain power up here and ask each of you to ask somebody else on the panel a question. Daniel, why don't you go ahead and start?
Yeah, Billy, I have a question for you. I think the work you've been doing in mapping the RNA editome is pivotal for us to kind of capture the opportunity of Axiomer and ADAR editing. You're obviously looking a lot at the natural substrate and the natural sequence context, and many of us work with chemically modified oligos. How much of that do you think will over time translate from the natural context to the chemically modified context?
Yeah, I think it's a great question. I would say the fact that now we're harnessing ADAR, we're already taking advantage of the nature. But I guess you're specifically asking about the substrate, the double-stranded substrate features, how much of that nature we can learn. I think the short answer to that is clearly yes. We can learn a lot from that. We actually have evidence to really demonstrate that. But of course, we also understand that chemical modifications is a very big part of the equation. So we're not saying that part only would be sufficient necessarily. So I think in the end, how do we really combine the chemistry and sequence is going to be a pretty complicated question, but also interesting one.
Yeah.
Ram, you want to ask a question?
I would actually ask the entire panel, only because we feel like, I don't know how others feel, there is this context around DNA editing and the amount of Cas systems and CRISPR systems that come out every quarter, so to speak now. that talk about doing more. But when you look at that universe, the number of single point mutations or genetic diseases you can go after, which is fantastic for those patients, is still a small subset. And my question to the panel is, what can we do to elevate what we're doing from an RNA editing standpoint in terms of the applications and provide a very cogent message relative to the large patient populations that we're going to go after, the large commercially feasible patient populations that we're going to go after over the next, I don't know, 5 to 10 years?
I think it's a great question. And I think, it's a great one because we can project outwards. Because I do think, I call it the shiny object, right? There's always this new platform and it yields like, here's a really cool thing, what can one do with it? But to your point, it really comes down to what are you gonna do with it? And I think as an industry, we've got a lot of shiny toys. The question really is about our industry's job is to make medicines. And if we think about ultimately our goal of what medicines are gonna come out of it, like read S1s, how many new technologies come out and you flip to kind of see what they're doing and it's like, it's the same 5 targets that everybody else is doing. And I think the opportunities that we really have in front of us with RNA editing is to open up and expand that target universe. It was something that I know we've invested in. It's a driver of our collaboration with GSK, and I think we're sitting at the precipice now of clinical genetic data sets that are opening up real unique opportunities where we can see a genetic insight of a target translate to a measurable outcome in a patient population that's tractable and open up big areas of biology. And I think RNA editing is perfectly suited to do that because the biggest concern one has when opening up a big area of biology is safety. Almost more than efficacy, it's how do you treat that in a way that's reversible, where you don't have to worry about permanent mutations in DNA and follow patients for a lifetime to see if maybe you've disrupted something. And so I think the opportunity we have to tackle big genetic data sets, to open up big targets that we can then tractably go after. We shared recently at the R&D day that when we looked at the tractable universe that we're using for what we like to call the editverse of RNA editing amenable targets using our application, right now through this algorithm that we built, you can target about 50% of the transcriptome. So if we think about 13,000 genes and continuing to expand that open up the opportunity to think much, much larger about targets, I think again, hopefully in a year we're talking on the panel beyond alpha-1 antitrypsin, beyond the targets that we're talking about today, but to medicines that are going to have massive impacts that disrupt not just DNA editing fields, but really think about other modalities.
Daniel, how would you respond?
Yeah, look, I think it's a good question, Rem. I think it's just about, we know where the target opportunities lie, right? We start to see where the opportunities are. There's many more beyond that, but it's now about just doing it. I think it's great for alpha-1 antitrypsin patients that all of you guys are testing this technology on that disease. But I think it's important that we look beyond that. And we're starting to venture there. Our first 2 targets are into more larger, more prevalent indications. And I think that's a good kind of test case to see how that goes and how we can capture that opportunity as well.
Paul, do you want to ask a question to somebody?
I'm happy to, but I think we've had a number. I mean, if I have time, I'm happy.
Go another minute or so.
All right,
throw
awesome.
Your question out there.
So I will take the same thought, which is I love to hear other opinions and ideas. But it is challenging, and you asked this question, Kay, but I think it's something that we all deal with when we talk about new modalities, new technologies, and real challenges that we face. And I find the fact that we're up here all working on unlocking this. I am curious about, various, what do you see? Do you see target challenges? Is it chemistry challenges? How do we get this? If we're really going to move to prevalent large diseases, what are some of the challenges still that we all face ahead of us that we need to address?
Want to go first, Ron?
I'm happy to, but I'd love to hear what Billy has to say as well. As I mentioned earlier, we're still looking through the window of what we can actually do here. And when you think about history, the modalities that actually succeed are when they have a good product market technology fit, right? I think folks in the audience will start asking, all right, so if DNA editing is there, why would you go after a rare genetic mutation, right? Alpha-1 is a perfect example. The patient population is very heterogeneous. There is a large variability in terms of mild, moderate, severe patients. Try convincing a doctor to do a mild patient and say, hey, you don't show any symptoms, but you have the Z allele and you're homozygous to it. Why don't we do a one-time permanent edit? Don't worry about it. You'll be fine for the rest of your life. I don't think payers are going to pay for that. I don't think physicians are going to sign up for that. Okay, so finding the right fit for the right patient, I think, is going to be important. I think the second aspect that at least I get asked is, well, why can't Alnylam and Ionis start doing this? Well, I think folks in the panel are still, and we've been working on this for at least a decade based off of the work at ProQR, and it's not necessarily easy, right? And so understanding where that sandbox is, understanding where that sweet spot is, at least for us at Koro, it's about activation of a certain protein. And this is what I mean by having the exposure and talking about what RNA editing can do. When you think about going from DNA to RNA to protein, right, you can actually have an impact, as Billy said, in terms of impact on methylation on DNA. You can make a single A to I edit on RNA and have an impact on methylation. You can have an impact on post-transcriptional modification that cause diversity of what the transcript can do. You have variant biology that talks about both coding and non-coding regions where you have G to A mutations that can increase expression, decrease expression, just from a copy number standpoint. There's a lot of evidence there in the genetics backs it. You can then think about impacting amino acids on proteins and create a variant of a protein that has an impact on biology. That's a large spectrum, right? And so you can think about which indication that specific modality is going to be best for. And in each of those cases, we're talking about activation rather than knocking down a specific aspect of the genome.
Billy, I started with you. I'm going to end with you. You have the final word on this.
Yeah, again, it's great to be on this panel. I think hopefully everyone's convinced. I think the potential of this ADAR modality is enormous. I think just the narrative of CRISPR obviously deserves a lot of attention, but ADAR, I would say, from a therapeutic perspective, probably has higher potential because of the feasibility and safety. So to address this question, so I think, to me, as a field, how do we? I almost want to ask a question, but I guess we don't have time to answer the question. How do we balance this kind of platform development and the assets indications? So clearly, we want to move both forward. So in the end, I think as a field, we really have to prove this platform is going to be viable, not for AATD, but also for many other indications where the true potential of this technology is. So I think balancing the platform technology and the indications is going to be a very challenging but also exciting task ahead of us.
Okay, well, very good. I want to thank all of you for this very informative and entertaining session.
Thank you,
Thank you, guys.
Appreciate it.
Yeah, thanks.